Hello everyone, let's talk data. I'm Vanessa Cave and with me is my colleague Salvador Gazan, and we are going to talk to you today about blocking. So, Salvador, it'd be great if you could give us a lowdown on what blocking actually is.
Oof, that's a very good question. We always we talk about the three elements of the experimental design, and one of them obviously is blocking. Um, I like to think about blocking as selecting homogeneous experimental units on which we are going to put all the copies of a set of treatments. So obviously, the most natural way to think about is a field experiment where we have, let's say, 20 different treatments that we're evaluating. We can think about varieties, and then obviously we want to replicate that several times, and we do them all together, group as a unit, which in this case will be a block or a replicate all together.
Yeah.
Um,
Yes, I guess I'm more familiar with blocking from animal trials. So in that kind of context, we could think of it as grouping animals together that are as similar as possible. So, for example, on age, on body weight, on body condition score, and then so with and these groups of animals that are as similar as possible, we call them blocks, and then the treatments are assigned to the animals within the blocks at random. Yeah. Yes, so your
in,
variation between experimental units, I you're trying to get that smaller within a block than it is for units between blocks. Does that sound about right to you, Salvador?
Yes, it sounds right. I mean, obviously, you that's the reason why you look for individuals, let's say that they have the same age or maybe the same size. And in plants, we tend to pick areas that are more or less compact, where we define the blocks, where we suspect that the nutrient or water availability is going to be the same. But on the same sense, we have different blockings in plants too. I mean, I've seen blocking when you have in a greenhouse, maybe some of them that are on the left or on the right, where we suspect maybe a change of temperature, which again is also something physical.
Mm.
But sometimes you have blocking also for size. If we have the plants growing before we plant them, we might say, okay,
Yeah.
we'll put all the little ones together, all
Yeah.
the bigger ones together. And then I tend to think about other blockings, such as like, for example, when you establish the experiment, you might say, okay, we did this experiment in the morning. We apply, let's say, some chemicals to some insects, and then you're going to say, okay, the morning is one block because we suspect something is happening in the morning. The afternoon is another block. So we define blocks on time, or sometimes even person. So
Yeah.
we have also a combination of person. So I'm sure you see something like that in animal too, no?
Yes, yes, we do. Yes. So I guess the purpose of blocking is really to even out that natural variation amongst experimental units, be those plants or be those animals, etc., and to make sure that the comparisons between treatments are as fair as possible. So really our blocking's based on most on known information, isn't it? That we have about our experimental units or suspected um, differences between them, be that like spatial in a field trial or something to do with the animal's health status in an animal's trial, in an animal
Uh,
trial.
and
Yeah.
it's pretty much anything we can suspect it might have something. I mean, we know from previous experience that, let's say, the field or the greenhouse has different behaviors in the front and the
Mm.
back. Um, but sometimes things that you suspect they might affect. I mean, that I'm not very sure, and you might even kind of test it as you do your
Mm.
experiment. And
Yes.
and and this is the other thing that is interesting because, and I have a question for you there. So let's say I have my 20 treatments, I have my blocking factor, I have 6 blocks on my experiment, and then I do my ANOVA analysis, and I find that my block is not significant. So should I drop it from my analysis? I know what your answer is, but I'm
Yes,
still
I would
asking you.
say a definite no, because your randomization is based on the restriction that you have these blocks. So that's one reason why I wouldn't. And the second reason is just because your hypothesis test provides no evidence that the block is important. It doesn't actually mean it isn't. Yeah, you just don't have enough. You may just not have enough power to detect it. So yeah, I would always keep those blocking variables in my analysis.
I totally agree with you. I mean, it's the price for that insurance that the blocking
Yes.
might bring you
Yes.
is that
Yeah.
loss of degrees of freedom,
Yeah.
which is the number of levels of the number of blocks minus one. And obviously, if you originally restricted your randomization, and that's how I like to see it, you restricted randomization to be all the treatments in one place, not just completely like a completely randomized design. You say, no, I want a copy of each of the treatment here, and that's a restriction. And the cost is those degrees of freedom.
Yeah.
And the other thing I tend to think about is if you were allowed to move everything everywhere, then your error variance will be a bit different because you are allowing to have the same treatment differently,
yeah.
not restricted.
Yes.
So
Yes. Yeah. Absolutely. So when we read like research papers, there's a few terms that come up. There's balanced, unbalanced design, there's complete, and there's incomplete block designs. I wonder if it's useful to have a chat about those. So maybe we could start with what's a complete block design as opposed to an incomplete block design.
well, that's a good question too. My typical experiments when I work in forestry, we have I might have 700 different clones or to test. And obviously then you think, oh, well, I have each clone replicated 6 times and I'm happy with that. And it's a big size of an experiment. But then when I look at for a block and it has 700 plants all together, it's an area that is too big.
Yeah.
First, logistically, it's probably a bit too big. But then second, you think, is this really homogeneous enough to be able to represent that all the treatments are more or less into a homogeneous section? And then you realize it is not, because you have pretty much each of those blocks is as big as it could be an experiment.
Yeah.
So that's when we still keep this replication, and then we use incomplete blocks to make smaller units within that big block of 700. And that's a type of design, which is a resolvable incomplete block design.
Yes, it's another term, resolvable.
Yeah, another term for resolvable, yes, because obviously you still have the big reps and then you have the incomplete blocks. And what I like to think about that one is, well, it's in strict rigor, it's not a block because you don't have a copy of all the treatment, and that's why we call it incomplete. But if it's a decent size that obviously you know about how the variability of the soil goes, you can think is going to be more or less homogeneous to define a block.
Yeah.
So
go
ahead.
I was just so if you can, is it best to create a design with complete blocks, or does it really come down to the nature of how you define your blocks and how you create your blocks? Perhaps that's a bit hard to answer.
Yeah, so I will always prefer to use incomplete blocks whenever I can. This is again working on field experiments. So even if I have, let's say, only 50 treatments, I will try to put incomplete blocks, because the role of the incomplete blocks in that case is going to be more than just creating these homogeneous units. Actually, what it's going to do is allow me to some form of spatial control. So like a spatial analysis. So because all the units will be higher or lower, it makes a little bit of a sort of microsite correction on the incomplete block. So most of the time I tend to recommend them. But you have to be careful that they're not too small or they are not too big. And that's where the challenge comes into place.
Yes.
And if they are too big, obviously they're not homogeneous enough and they're not representing what homogeneous or a unit you want. If they are too small, you run another risk. And the risk is, for example, if you have an incomplete block of size 4, and 2 of your treatments have very high values just by a random chance, they're going to push the mean of the incomplete block high. And that means it's going to be corrected, that block as an effect, which means it's going to extract part of the signal of those treatments. And then you have increased variability on terms of the incomplete blocks, but you have reduced variability on the treatment effects. So too small will not be necessarily very good.
Yes, indeed. That wouldn't be good at all. So it's a little bit, I guess, when I think about animal trials, it's a little bit different because we tend to have a lot, just a few treatments that we're interested in, and lots of potential animals to recruit into the study, which are our experimental units. So there's a little bit more flexibility in ensuring that you design blocks of animals which are as similar as possible.
We actually, in aquaculture, I have seen incomplete blocks defined by the tanks. So obviously the tanks, if you have, let's say you have replications and you have, I don't know, 16 families that you want to test, then sometimes the tanks, because you need large number of individuals per family and so on, so it might be that the tanks are not big enough to hold all the families, so you can have incomplete blocks in the tanks. And sometimes the imbalance occurs just because some families, individuals die and so on, but it's not part of the design. And actually, one thing we didn't mention too much, but I think is relevant, it's the 2-way or even 3-way blocking.
Yes.
Do you see, I mean, do you have an example of that, or do you, how often do you see it?
Yes, I obviously do less plant breeding trials than you do, but it's occasional glasshouse trials where things are laid out in a rectangular array. So we're blocking there by rows and columns. More commonly, I guess, using type of a nested blocking structure like a split plot design in some of our experiments where we're testing interested in 2 treatments, and we've got one which is easier to apply to smaller plots, but one which you have to apply to a bigger plot. So a good example of this is it's easier to apply fertilizer to a bigger plot, but cultivar to a smaller plot. So that's another type of nested blocking structure. It's a bit different from the row column one.
I mentioned in entomology, which I also have work, it is common to have the 2-way blocking between, actually even a 3-way blocking, because you might evaluate let's say you're evaluating mosquitoes for some form of repellency, and you are, let's say, trying different chemicals. The chemicals are prepared on different days, and the study is done in different days, but also there's different staff preparing the chemicals. So you might have that 2-way blocking between the day and the staff. And then even sometimes you have another blocking that goes with, maybe you're dealing with different lines of mosquitoes. So it gets quickly a bit more complicated.
Yes,
the analysis is not really that complicated.
No,
but it is a bit more complicated when you plan it.
Yes.
And actually, I know I'm jumping around, and I'll ask you, we talk about incomplete designs, but we didn't talk about balance and unbalance incomplete block designs.
No, we didn't. No, yeah. Yes. So let's. I know you mentioned balance earlier on. Yeah. So this is a concept that many people, well, it can be quite confusing in a way. So one aspect we have, of course, is having treatments replicated the same number of times and uh, kind of spread across the different blocks. So the treatment compared, so the treatments occur in the same block an equal number of times. We can have other designs where this is not the case, and we have the issue that then the treatments and blocks aren't orthogonal to one another. Yeah.
I like to think about also that, because the balance designs that, like you say, they want to be fair to compare every treatment against every other treatment pretty much the same number of times,
Yeah.
are good because they're very fair to compare treatments. And
Yes.
somebody will say they are going to make the standard error of the difference between treatment pairs all equal, so all
Yeah.
fair. But they are very hard to come up with, in the sense that they're very restrictive. They only occur, when you look at the books, they only occur for certain combinations of number of treatments, replications, and so it's not very easy, and size of the incomplete block. But the ones that are unbalanced, obviously it's just more flexible, and you can have all those possible numbers.
Yes.
But it's still more risky, because then you have some treatments that are going to be compared more often. They're going to be in the same incomplete block more often, and therefore they're going to have lower standard error of the difference, and so on. So you have a kind of like a challenge of
bit of a trade-off. Yeah.
But I just need to mention that the on breeding trials, normally it is not too much relevant to have unbalanced incomplete block designs, because our objective is not too much to compare the treatments with let's say a t-test or some sort of, it's more about just estimating the effects of the different genotypes.
So you're more
there's a
interested
bit
in
more
ranking,
room.
are you?
Yeah, it's more about a ranking. So that unbalance, it's not ideal, but it's actually just very manageable.
Yes.
And not so much in the inference. But anyway, so
Yeah.
it's
That's
just
a good point. And again, I think with animal trials, they tend to fewer treatments, and it's a lot easier to design it so you've got a balanced design. Really. Yeah.
exactly, and it's safer.
Smaller number of treatments makes it easier. And we have flexibility over how many blocks you have and the block sizes. Yeah. So talking about blocks and block sizes, what's your kind of thoughts about how many blocks there should be in an experiment? Like, is it sensible just to have 2 blocks, or?
So obviously, if you can have, I mean, it all goes into the replications of your treatments. So I will say, I mean, that then the question is obviously how many replications, and then you will put it as blocks. I will say, if you don't have enough replications, always try to do it as blocks. I mean, even if you have 2 blocks.
Yes.
Yeah. Now, if you're going to have many more replications, I will try to pick as many blocks as I can have. And actually, I can provide an example. So there was one crop. This was on cocoa. So we have lots of clones of the same plants to put into a field. and we have up to 50 plants per family, per clone in this case. And it was a couple of 100 clones, so this was a very large experiment. And we have the decision, do you put 50 blocks, or do you put one, or you put one big plot with all the 50 plants together?
Oh,
Obviously,
okay.
one big plot with all the plants together is not ideal because you have no replication at the end of the day, and all these 50 plants are growing in one end of the field or in the other end, so you have no way to control and compare them very fairly. But they're growing as a crop, as a group, which is a bit more how you're going to grow them and commercially. On the other hand, you have 50 reps with one copy of each plant all together and randomized, so you get the maximum number of degrees of freedom you can get, and you can have every clone represented 50 times on different areas on the field. So it sounds like ideal. But then you have to think about the competition between the plants, because sometimes some plants are more vigorous than others, and they have some induced competition on the other plant. And obviously you are competing with a variety that, or a clone that competes with another clone in a different way. I'm going a bit long, but so then in that case you think, well, is that the answer, having 50 replicates? And the answer is no, because you have to think about what you want to try to mimic.
Yeah.
And in this particular study, we decided to use small plots. So let's say if you have plots of 5 plants, all in a row, then what you have is you have 10 replicates, which is still a decent number, and you have enough degrees of freedom, and you can check the degrees of freedom, but at least they are growing into a row. You can also have 5 replicates of 10 plants, let's say 5 by 2, which mimics a bit more of a plantation. So the question there, and this is answering your question, is my question goes to the actual scientist, and I ask him, what is the minimum size of your plot that you feel comfortable with?
Okay.
And in this particular study, they say 2 plants.
Okay.
So we put 2 plants together of the same clone, one next to each other. We know they are competing with each other and they have other competitors, and you're able to see them a little bit better. And then we had 25 replicates, which is it's more than enough.
Yeah.
Okay.
Oh, that's nice.
So that's
nice.
the long story of what we did a while ago.
You are listening to Let's Talk Data, brought to you by VSN, a world leader in the advancement of big data software solutions for the agriculture and bioscience sector. Let's talk data.
So another question that gets commonly asked and discussed a lot is whether the blocking factor should be a fixed or a random term. Now, I'm really keen to hear your thoughts on this. I know that at times I differ from my colleagues in how I think it should be treated, but yeah, so really keen to hear what you think.
That's always the question.
Yes.
I was afraid you were going to ask for that.
I thought I'd ask you before you asked me.
Yes. So the answer is it depends. And I do have a few rules, and I normally explain this, but so first we need to get into the context, the historical context that I like to put myself in. We learn when we learn our statistics, and we still, with many of the books, we see blocks always as a fixed effect, because originally we were only fitting linear models. And it makes sense. Blocks were fixed effects, and there are nuisance effects that you just control for. And we still do it. Now, I like to rethink that process when we write down our model and say, well, are they really fixed or not? And remember, when something is a fixed effect, we're saying these are the only conditions I want to base my inference on. And that's where I'm like, do you really only care about those blocks, not other blocks in the world? which in reality means actually no, because if you're finding a treatment, let's say a new fertilizer, you're going to use that fertilizer somewhere else for other blocks, not for the same blocks. So it seems a bit restrictive. Now, if you're doing another type of a study, and it happens to be that it was Monday and Tuesday when you evaluated those things, and those are the conditions or specific batches that you had, then you can think about, this is not going to go to any other batch. Sadly, I just had what I had. And then you can think about it's fixed. But in reality, in field plants, I tend to think like, well, could be anything. So I like to think they should be random.
Yes.
And then I tend to pick them random every time I can. However,
yeah, there's a however.
Then you have to start thinking about, do I have enough degrees of freedom to estimate the variance associated with those?
Yes, yeah.
So that's the first thing. And then if you think, oh, I have, like we talk about a case where we have 2 blocks. I mean, if it's random, then that means you have 1 degree of freedom to estimate that variance, which can go very high or very low, which is going to make all your inferences to be all over the place.
Yeah.
So in that case, you don't really have information to estimate it as a random effect. You're better off to get it as a fixed effect. That's my logic. But then there is another thing that sometimes I tend to do, and this is a bit of a dirty trick. Is my model too complex?
Right.
So for example, if I'm analyzing several trials together, or maybe a bivariate analysis of some sort, and if I make the blocks as random, then I have to have more variance components to estimate for each trial, or some correlation between traits if I'm doing a bivariate analysis, and the model is more likely to kind of like not fit or crash or require more attention. And in reality, I don't really care too much about blocks. They just have to be there for correcting. So then I tend to use them in that case as a fixed effect, sort of like, oh yeah, that'll be okay. It won't create too much of an issue.
Okay. So you do that in preference to doing a meta-analysis on the summary results from the individual trials?
Well, we analyze
or is that another approach
together.
to do, maybe?
We analyze everything together in multi-environmental trials. So just from the raw data originally, and then it just blocks are just a nuisance effect.
Yeah.
To be corrected for. So how about you then? I have to.
Yeah. Well, I guess coming from kind of a design background where, as I said, we had kind of mostly simple balanced complete block designs with a sufficient number of blocks, and could be analysed in the ANOVA framework, which makes everything very nice. And then I guess I kind of took that concept on when I had to move into a linear mixed model kind of REMOR framework for more complicated data. Perhaps it was unbalanced and things. Yes, I tend, the majority of the time, if I can, view my blocking effects as random. They're kind of nuisance variables. They're a random selection of possible blocking variables that I could have, and I do want to generalize my results beyond the blocks that I just happen to have observed. And again, there's some exceptions. Sometimes, yeah, as you said, if you don't have very many blocks, my rule of thumb, that's sort of a rule of thumb, is if I've got fewer than 5 blocks, it's not worth even trying to fit it as a random effect. Simply just don't have enough information to estimate that block variance component. Yeah, so yeah, that's where I'm at.
That's good. I just came to my mind 2 concepts that I always had a hard time. When we consider blocks random, we assume there is a normal distribution, of block effects, and we are sampling from a population. But in reality, I have a hard time thinking we're sampling from a population, because if you think about the blocks, particularly in the field, they are next to each other. It's not like we have multiple possibilities. Yes, if we move the experiment a bit to the left, a bit to the right,
slightly
they
different.
will be slightly different,
Mm.
but they are actually almost like they are a sample of blocks, but they are kind of correlated if you think
Yes.
about it.
Yeah.
You know, so, and you want to try to infer about a population of blocks, but you're not giving yourself the chance that the blocks are everywhere, like the way we think about like a random treatment effect. So that I always find that it's an interesting concept. And I don't have an answer for it. I think it's just part of the assumption.
Yeah.
However, the other thing I tend to think about incomplete random blocks, which I like it, is when I have unbalance or unbalanced designs.
Yeah.
And normally when I do incomplete block designs, I always use random effects of incomplete blocks because I don't have a copy of all the treatments. And I know that the idea is when it's random is that you use some recovery of information of the intra-block and inter-block and so on. And the mathematics are not totally clear to me, but what I like to think about considering incomplete blocks as random is that you're doing a correction, but because it's a random effect, the correction of that block is not as strong as if it was a fixed effect because of that shrinkage.
Yes. Yeah.
And if you remember when I mentioned if you have only 4 genotypes in one block and you're going to correct all 4 of them, if you use them as fixed, you might overcorrect them too much. If you use them at random, yes, you're doing a correction. It's still a bit too much of a correction, but then because it's random, it's not going to be too bad.
Yes.
And again, it goes with the concept of a bit of a spatial control on the incomplete block. So yeah.
Yeah.
So,
I guess it kind of also highlights that when we're designing an experiment, be it a incomplete block design or whatever type of experiment, we have to have a little think about how we're going to analyze the data once it's collected to make sure that we have got sufficient information to do what we're proposing to do.
And actually that brings another thing that somebody asked me not long ago, which is about the tiers of blocking.
Okay.
Have you heard about that concept of?
No, I don't think I have.
It's when, for example, it's it's a bit like a nested blocking. Well, it's hard to define it. So the best example I can come up with, if you are, let's say you're growing wheat, different varieties of wheat on the field, and you have, let's say again, 6 replications and so on, and then you collect the wheat, and then you're going to process to make bread and assess the quality of the bread or the protein content and so on. And then you take it to the lab and you have your samples from the different replicates on the field. And now you have to do again more blocking because you can't process all the batches at the same time.
Yes.
It's the tier.
Yeah, or sequential or
sequential blocking. Or,
I
can't remember the actually. Do you find something like that in your field?
To be honest, I haven't come across it a lot. No, to be honest.
In my case, I have. In my case, I have. But so what were you saying?
Oh, so sometimes it happens, say, when the data is being collected, that it's only possible to measure a certain number in a day. So then we would recommend that blocks are measured like within a day, maybe 1 or 2 blocks within a day, rather than measuring across blocks. Yeah.
So
But no, I
that's a
haven't come
good
across
that's a
one.
good that's a good case, because like
Yeah.
if we talk about the same wheat experiment and you're only able to measure, let's say, 2 blocks in a
Yeah.
day, the amount of experimental units for 2 blocks in a day, do you do it across all the blocks, or do you do it in just 2 of the blocks, and
Yeah.
then next day you do 2 other ones, or you do 1/3 across and 1/3 and 1/3? And that's interesting, because when you do it in the 2 blocks, you're confounding the effect
Yes.
of the day you're measuring, or the crew, or the person who is measuring, or whatever other effect. You're confounding that with the blocks on the field. And that might be good or bad, because in one sense you're adding another kind of like a error or the random term. And then you might not, whenever you do the analysis, you just say, okay, well, I'm not being able to separate what is the true effect. I think it's better when we think about the case that you can only do 1 replicate a day.
Yeah.
So if you do 1 replicate a day, the effect of that replicate gets confounded with what you're doing that day, or how the conditions for that day are. And yeah, so I'm suspecting that the variance of the block might be increased, because you're adding another little term. Yes. And it's not going to represent the same physical thing that you thought it represents. But if you do it the other way, which is actually doing it across, doing 1/6 every day, you have to design the experiment in a way that you have the 2-way blocking.
Yes.
And that means you also want it to be balanced.
Yeah.
Or orthogonal to the treatment. So you want every treatment to be measured on every day across the 6 blocks.
Yeah.
Which obviously means now you have to have it a bit differently arranged. And so you need other software to design. But the advantage of that is that when you do the analysis, now you have 2 sources of variability that you can assess. Oh, yeah, the day variability is different than the block variability, and which one is bigger, and so on.
Yeah. So this must be quite a challenge in some of these big field trials, particularly when you've got like a row column design and you want to do a spatial analysis, and then you've already got the row the column blocking structure going on. You're trying to model the spatial variation across the field, and over and above that, now you've got this extra difficulty that you can't measure the entire field all at once. So you have to plan that. Yeah.
And then also that's without counting that maybe you take the samples to, like I mentioned before, to the lab, and then you analyze them in the lab, and you might have another layer. So obviously the best solution in those cases is just to allow for the confounding. Treat each block as a unit that you're going to analyze together, measure together, harvest together, and process together in the lab, and that's the end of the story. Otherwise, you have to sit down with somebody and try to make your design as balanced as possible, and so on. So now there's always the safety net, which is not always possible, that if you randomize, then you will get rid of the problem. So obviously you can randomize when you measure in the field, but you can randomize in the lab and say, okay, I'm just going to process 1/6 of the samples I have today. I will just mix them up and pick some at random, obviously, and then you probably are going to get a copy of each treatment and so on, and it doesn't matter.
So, that's true.
but that's good. Yeah, no, that's another case. So we talk a lot about the different types of blocking, fixed and random, and also how we use them on the spatial analysis. And then obviously this confounding and so on. So quite a bit of different topics.
Lots of interesting nuances and things to think about when you're designing an experiment and when you're analysing the data. Yeah, it's a fascinating topic, experimental design.
Well, Vanessa, it was nice talking to you today.
Pleasure talking to you.
We'll meet next time.
Yes.
And keep having fun with your data.
I will, and you too. Thanks everyone for listening.
You have been listening to Let's Talk Data.